Thank you, Yavuz, and thank you, Dilip, for the invitation. And I'll start presenting. I'll share my screen now and start presenting my talk on in silico drug discovery, in silico discovery of multi-targeting drug candidates. Can everybody hear me? Is that okay?
You are perfect, Amiram. We can hear you very well, no worries. Feel free to continue. And at the end, we will have question and answers. The floor is yours, dear Amiram.
Do you see my screen now?
Yes, I can see your screen, but you need to click. It was bigger, now it is smaller. You need to click on the webinar, I think. You cannot see the full screen.
Here it is.
Now we can see your screen. Perfect.
Good. Good.
And your voice is amazing. So please go ahead. Thank you.
Thank you very much. Many of you are probably aware of the fact that many molecules that are introduced as drugs are not directed to a single target. But even the attempts to direct a molecule, a drug candidate to a single target, ends up with hitting many other targets. Now, this could be a problem of creating health problems and side effects, and it could also be used as a tool in order to improve the therapeutic outcome. So what you see on my front slide is a few. I'm trying now to pick. Okay, just a second. Spotlight. Okay, here's. Okay, can you see my spotlight now? Is it moving around? Okay, so a single molecule can interact with several targets, biotargets, as shown here. On the left, it's just a cartoon to show that parts of this rooster molecule can hit several receptors depending on what part of that, what fragment of that molecule hits each of the receptors. But there are also quite a few known examples. I will not show many of them, but this is one pretty well known, which is used as a candidate against Alzheimer's disease, which hits both acetylcholinesterase and butyrylcholinesterase, and base 1, which is the main culprit in cleaving the amyloid polyprotein precursor, and also affects beta amyloid aggregation, probably through its interaction with the peripheral site of acetylcholinesterase. Now, the concept of drug discovery has gone through quite a few stages in the last 50 years or more, until about the mid 80s and maybe even later. The main paradigm was that one disease has one target and requires one drug with high potency and selectivity, but that was transformed later, actually in the mid 80s with HIV especially, that one disease has many targets and each target requires a drug. So it ends up with many drugs, and each of them has high potency. Each of them should have high potency and selectivity. And that was what we used to call a drug cocktail, and still call it today. The new paradigm, which is gaining momentum over the last decade or so, is that one disease that has many targets requires probably one drug, which is much better than having many drugs. So that drug may have lesser potency and selectivity, but would be much more effective finally. Most of the diseases that we are dealing with have been or are still treated by what is called combination therapy or drug cocktails. And those are multifactorial diseases which have a few pathways to the development of the disease. And that is true with cancers, with Alzheimer's disease, diabetes, nervous system disorders, hypertension, cardiovascular diseases, many or most viral, bacterial, and fungal and parasite infections, immune system activation, obesity, and so forth. And the idea of multi-targeting is now gaining momentum because most researchers realize that it is much better than the combination therapy. So combination therapy apparently deals with all the different targets in a specific disease, but a single molecule that can provide the same kind of interactions that a few molecules can provide. A single molecule has just single molecule pharmacokinetics. It reduces the risks for drug-drug interactions. It has less side effects and less toxicity, less chance of resistance due to mutations, simpler dosing and patient compliance, and easier regulatory approval process. So now that I hope that you are convinced that we need single molecules that will be multi-targeting, how do we achieve it? The one way to do it is to pick two known drugs, each of them hitting one target, and combine them with a chemical linker, such as a carbon chain or a peptide chain. And as you see here, the combination of chlorotacrin and Trolox as a heterodimer, the difficulty with that kind of combination is that it increases molecular weight considerably combined to a single drug, and it also creates problems from the side of hydrophobicity. So both hydrophobic and molecular weight issues are involved when we use linking. The other type of combination is fusing the two molecules. That requires some special synthetic procedure. It is sometimes possible and some other times it is not. As you see here, you use that nitrogen in order to combine those two molecules into one. And again, now the molecular weight is approximately that of the combined two molecules, but the molecular weight is of course larger than of simple smaller molecules on one hand, and the hydrophobic effect is also still prevailing in that kind of combination. The best possible combination is to merge two molecules into one. That is possible only in very, very few instances. And the one that I'm showing here is one that came from two Israeli universities, one from the Hebrew University in Jerusalem, the rivastigmine, which was invented in the lab of Marta Weinstock-Rosin, and rasagiline, which is both of them are used drugs. This is a MAO-B inhibitor. Rivastigmine is an acetylcholinesterase inhibitor that was on the market for many years and still is. Rasagiline is a MAO-B inhibitor that is used as a Parkinson drug, and both those molecules were fused or merged, actually, not fused, merged into this ladostigil, which is still in clinical studies for treating originally for treating Alzheimer. Now it is for treating several other disease conditions. The idea here is that ingeniously the chemistry of those two compounds could be combined, and since the triple bond here is the one that is responsible for irreversibly binding to MAO-B, while the rivastigmine part is the carbamate, which is this those 4 atoms over here, which block the serine, the active serine at the catalytic center of acetylcholinesterase. So this is a probably unique example of such merging of molecules. I am not sure that there's any other such merging that was successfully brought to clinical studies. So all those examples were from chemical synthesis, while we are interested in trying to do the same by rational design. So rational design could start by querying, for instance, which structures are similar to a drug combination. So one could take the 2 drugs, such as ladostigil here, and find out which through SciFinder, through the similarity programming of SciFinder, one could find which molecules and how many of those are closely related by chemical similarity. So that is one way, but the downside of that is that we will get more of the same. So if we're looking for scaffold changes, or what is called scaffold hopping, then this is not the best way to do it. So we have a different way of dealing with that, and I'm trying now to present how we deal with the issue of in silico discovery of multi-targeted drugs. So we start with single drugs, and the way that we do it is that we query which properties could distinguish between molecules that are bioactive on a specific target compared to other molecules that cannot hit that target. So most of the world of chemistry, most of the chemical space would be neutral to that target, and there will be a few molecules, maybe a few hundreds, in some cases a few thousands, which are binding and affecting a specific target. Affecting, it could be inhibiting, it could be agonistic, it could be antagonistic. We look at a specific property, at a specific biological property that is related to a specific target. And the difference that we are looking for is not a difference in structure, we are far away from similarity or from looking for similarity, but we are only focusing on physical-chemical properties. Physical-chemical properties that distinguish the active molecules from the inactive or the neutral molecules. And we examine those differences on 2 levels. One is how different are, or which properties distinguish the actives from the rest of the world of chemistry. And the second level, which is more focused and allows us to look for leads rather than just hits, as I will show soon, which properties distinguish the best molecules from the worst ones. The best are the ones that have the lowest IC50 or the best EC50 and so forth. So what we do is that we transform the molecular properties to numbers for both the actives and the inactives which we pick randomly from a huge database of molecules and dilute the actives sometimes 100-fold and sometimes even 1,000-fold. So for each active molecule, we have at least 100 molecules that are not known to be active. So this is an attempt to simulate the idea that among the world of chemistry, only a few molecules will be able to hit a target and not, it is not something that can be compared between a group of 100 molecules of actives versus 100 molecules of inactives, because the main idea is that we are going to build models based on learning from a very diluted group of actives by inactives, and then apply that model to the world of chemistry. So we represent the molecules mathematically in order to relate their properties to their activities. And we're talking about properties such as lipophilic character, volume of molecules, topology, charge, dimensions, and so forth. And we start with the group of molecules for learning, including, I remind you, including both actives and many, many inactives. And we know the activity of each of them. The activity of the actives is assigned by 1, and all the rest, most of them, have 0 activity. And then we transform the structure of those molecules to a set of properties. And we use about 200 properties. We use the program called MO by the Canadian Computing Group. And once we have the numbers that represent the properties of each and every molecule, we are trying to build a model that associates between those numbers and the molecular activity. More precisely, which properties are the ones that determine the activity or the inactivity. And that would be finally bring us to a computer model which associates the properties with the activity. Once we have that model, we can now move on to the big, to the larger, much larger chemical space where we have molecules with unknown activities, and we pass those molecules, we transform those molecules also into their properties. We pass them through the computer model. I'll show in a minute that we're using some kind of filters of properties, and now we can predict the molecular activity. So that's the main, that's just an outline of the idea of how we do that. Each molecule of the learning set, as I said, has about 200 properties. The input is the SMILES notation. The program calculates through the SMILES notation, the program calculates all those 200 properties. And once we have those properties and we know the activities of all those molecules, we try to produce filters, each filter of about 4 to 5 properties, where we search the most crucial properties that distinguish between actives and the randoms, as I told you, diluted either 1 to 100 or 1 to 1,000. If we're talking about the filters of 5 properties picked randomly out of 200 properties, we're talking about 300 billion picks. So obviously this is a very time-consuming issue, and even on a very strong CPU with lots of parallel processing, it could take months or even more to perform all the possible sets of 5 out of 200. So we devised this algorithm to shorten the search. And the algorithm starts with this combinatorial space of 200 or 180 properties. We build filters randomly. We pick randomly filters of 5 properties. This is done by random sampling. We check the ability of each filter to distinguish between the actives and the inactives. And once we made our mind upon the properties that are not appropriate for distinguishing between the two sets, we eliminate those properties and we end up with a second iteration where we have less properties to deal with, and we reduce the number of properties in several iterations, where we eliminate more and more properties. So that's why it's called iterative elimination. And we reach the final step where we have only about 1 million combinations possible, which we call the exhaustive search. We calculate all the 1 million filters. We order them by their ability to separate between, to identify correctly actives and inactives. And that is going to be the basis of our model. So iterative stochastic elimination, and for short, it's ISE, decides to eliminate properties that consistently contribute to the worst filters. ISE constructs the best ensemble of filters to distinguish between two activity classes on a specific target. It orders the filters, as I said, by their classification ability. A model for a target is a large set of filters. It could be hundreds of filters. It could, in some cases, it is even thousands of filters. Each filter would be composed of a set of 5 different properties, or rather ranges of properties, such as molecular weight between 350 to 415. Lipophilicity between -3 to +2, and so forth and so forth. So those are the filters that we have. Those are the properties that we have in each filter, and each of them has a quality which is, in short, it's called MCC. This is what we call the Matthews correlation coefficient. Matthews correlation coefficient allows to ask the question whether a certain filter, to what extent a certain filter is able to distinguish between the positives and the negatives, or the actives and the inactives. And the statistics for each model includes the Matthews correlation coefficient, both for the best filter and for the average filters, the ROC curve, the area under the curve, enrichment factor, and others. And if it's a good model, if we get a good model by the statistic criteria, then we can move to the next step, which is the screening of millions and millions of molecules. So as I told you, we start with maybe a few hundred actives versus many thousands of inactives. We create the model, and now we are ready. If a model is statistically sound, we move to the next step, which is the screening of many millions of molecules. We are talking usually about between 2 and 20 million molecules. Now we recently have the ability to look at 160 to 180 million molecules, which we can screen and either purchase or synthesize in a very short process of a few 3 to 4 weeks. So in each of our models, we are now able to run many millions of molecules. And the way that we do it is each molecule with its properties has to pass through the filters of the model. I represented here just a few filters. So the MCC of the best filter is 0.67, the next one is 0.65, and so forth. And a good molecule will be scored, all molecules will be scored by their ability to pass the filters either directly or not pass the filters. The weight that they will get is from the filters that they pass. They will accumulate the numbers of the MCC from the filters that they pass, and they will lose the numbers if they do not pass. So let's look at that molecule, and all the numbers will be between -1 and +1. So this good molecule passes through most of the filters, is not able to pass 1, 2, 3 other filters, and it gets a score of something around 0.6 or 0.7. And a bad molecule, an inactive molecule, should hopefully only be able to pass very few filters, miss most of the filters, and would get a negative number because we deduct the numbers, the numbers of filters that it does not pass from the total. To show the process in a more schematic way, so we start with the learning set of actives versus inactives. We transform the molecular structures into molecular properties. We know the biological activities of all the molecules. We introduce them into this algorithmic engine. Target 2, and among those we will pick the molecules that we will send to our experimental collaborators. All our projects are from the beginning are associated with a group that can do either in vitro studies or animal studies and so forth. So here's just another scheme showing that we start the multi-targeting by passing a multitude of molecules, many millions of molecules through target 1, then through target 2, then through target 3, which could be also an anti-target. So anti-targets are also part of our thinking about any project that deals with potential therapeutics. We would like to find out from the beginning what anti-targets we have to avoid, and if we want our molecules to have positive numbers, positive scores for model A and model B, which are targets, we would like to have a very negative result for those molecules, a negative score for the anti-targets. And we usually, in recent years, we worked mostly with about 20 million molecules from the zinc library of the University of California in San Francisco. We end up with about 50 to 100 molecules. Those are molecules, the molecules that we send, first of all, to our solubility studies, experimental solubility, kinetic solubility, and then those that are soluble are sent to our partners. So what I'd like, how much more time do I have?
You have another 15 minutes.
15 minutes, that's a lot. Okay.
You're doing great. You're doing amazing.
Thank you. Okay, so I'll show you just one example, one of many examples that we have in recent years, and that is a modeling GPCRs of the central nervous system. This was the main objective of recent PhD thesis of Dr. Shaima El-Ataune in my group. And what Shaima did is to construct ISE activity models for agonists and antagonists of each of about 30 receptors, which are part of 6 families, the 6 main families of GPCRs in the central nervous system. The idea was to construct those models in order to both find novel multi-targeting molecules by screening. So if we have models for 30 receptors and both for agonists and antagonists, that's about 60 models altogether. And we can pass, once we have those models, we can pass millions of molecules and find out which, where would they be potential hitters, either as agonist or antagonist, and sometimes agonist at one receptor and antagonist in another one. The other very well-known idea in COVID times is to repurpose FDA-approved drugs. And when we deal with repurposing, we use usually the drug bank molecules from the Canadian group, which have about between 14 and 15,000 molecules that are both clinical or investigational, which is clinical in the beginning clinical phases, or advanced experimental molecules. The other objective was to understand the reported side effects of many of CNS drugs, or most of CNS drugs, to predict mechanisms of action and potential side effects of drug candidates, and to develop better understanding of the mechanism of CNS disorders. And the chapters in the thesis of Shaima, which was a wonderful thesis, those are the titles actually of each of the main chapters of her thesis, which was a very philosophical thesis, because most of it was to analyze, most of it was consecrated to analysis of the results. So all these objectives require screening by 31 receptor models ×2, because it's agonism and antagonism. And we looked at the cannabinoid 2 receptors and the histamine and dopamine, muscarine receptors, opiates, and the 12 serotonin main receptors. And altogether, we could not model because of lack of some ligands to some of the receptors. We only managed finally to have about 58 models out of the 62 that we were hoping to have. And one of the most interesting ones was to focus on the endocannabinoid system with the CB1 and CB2 receptors, because we are part of a cannabinoid center at the Hebrew University. Hebrew University is a central university for developing the ideas of the cannabinoid activity, mostly with my PhD instructor, Professor Rafael Meshulam, who discovered the tetrahydrocannabinol and then discovered the anandamide, which is the internal cannabinoid ligand. And the endocannabinoid system has those two CB1 and CB2 receptors. And when they are in the central nervous system, both of them are quite problematic in the central nervous system, because agonists of CB1 receptor create psychoactive reactions, while the antagonists create the opposite reaction and suicidal thoughts and so forth. But CB1 receptor is, among other factors, associated with obesity. And this is the work that we did together with Professor Joseph Tam, also in our School of Pharmacy. What Joseph Tam asked us is to discover which molecules could be CB1 receptor antagonist, but they should be also peripheral. So what we did, we constructed the model for CB1 antagonism. This is general antagonism, both CNS and peripheral. And then we passed through that model the Enamine database. That's a company that we are working with for many years, and the great advantage is that they supply their molecules within 3 to 4 days. The molecules are already on our table or on our collaborators' table. We applied the CB1 antagonist model and found about 500 or close to 500 candidates. But when we applied the properties that we wish to have for avoiding CNS, which is log P greater than, calculated log P greater than 4 in molecular weight, and the polar surface area and the number of hydrogen bonding donors, we remain with only 34 candidates. Out of them, we could purchase only 15 molecules. Out of the 15, 14 were tested and 7 were found to be active. So the activities were between 66 nanomolar to 3.4 micromolar. So some of them were inverse agonists, some of them were pure antagonists, and overall, we managed, out of beginning with more than 2 million molecules, to end up with 7 actives out of 14 tested. This was published some time ago in the Journal of Chemical Information and Modeling. This is the process, 2 million to 500 to 15 to 10. And now we're looking again into those 500 because we found that about 25 to 30 known drugs do not, known peripheral drugs, do not require all those peripheral criteria, which were very strict. So we allowed those molecules to obey only 3 out of those 4, and sometimes only 2 out of those 4. So we have 62 other molecules that are now tested, first of all, for their antagonism on CB1 receptor. Then they're tested in mice for their effect, for testing whether they are peripheral or could reach the central nervous system through the brains of those mice, and also tested on obesity models for the other effects, for the anti-obesity effects. One more test for those molecules, for those 15 molecules, was to pass all those 15 through those 58 models for Gpcrs. Those are the serotonin receptors, the dopamine, histamine, opiates, muscarine receptors. And as you may see, each line here relates to one single molecule's candidate interaction or possible interactions with all those 58 receptors. And it is clearly seen that only the CB1 antagonist model is available for all of them, while there are a few, such as SH5, which has both properties of CB1 antagonism and CB1 agonism, and a few less interactions all over. All those 50 molecules, by and large, interact mostly with the CB1 and CB2 receptors, and very rarely with any of the other receptors. So here we are covering both the side effects of those molecules, and if we pass known molecules through all those GPCR models, we can also deal with, and that was done largely in the thesis of Shaima, where she dealt with side effects of many drugs with those different receptors. The next question that we asked is whether we could look for multi-targeting, not only in the sense of passing through other models as anti-targets, which was the last slide that I've shown you, but whether we could find interactions of molecules of our CB1 antagonists with other associated receptors with obesity. And for that, we used the phylogenetic map of another professor at the Hebrew University, Yuval Tabach, who suggested that we look at all those genes which are associated in his phylogenetic profiling with obesity. So we looked at the products and the gene products and the proteins of each and every one of those genes, and made a list of which of those genes has either a protein product that with a known structure, which we could use for structure-based design, or it has known ligands for which we could use our ISE algorithm to construct a model and look for molecules that would be both CB1 receptor antagonists and agonist or antagonist for each and every one of those. So we found, here's a small list of some of the ones that we found, some of them with antagonism, some of them with activation, and we wanted to decide which of the protein products may be important and whether it should be inhibited or activated. The question of inhibition activation was directed to our pharmacological associates. They suggested that we look for, among others, that we look for, where is that list? Okay, I don't see it here. So that we look for a list of antagonism and agonism for some of those molecules for which we have information. We did that, and we found mostly that the combination of CB1 receptor antagonism with PPAR delta agonism could be beneficial for treating obesity, indeed
Dr. Goldblum, I apologize, but if we can wrap up in terms of timing, thank you so much. It's incredible. I don't want to stop the amazing speech you're giving, but we're limited in time. So if you can wrap up, I would appreciate it. Thank you.
So I'll just summarize, if I may. I'll just summarize the idea of this algorithm that in our lab is the basis for all research in recent years and the discovery of more than 70 different active molecules that are further processed, that are patented and further processed now. So ISE is our basis for discovering both effective and novel multi-targeted molecules. It provides large sets of bioactive candidates. It discovers new scaffolds and produces highly diverse sets. More than 99% of the best candidates have never been described in literature or patents. It minimizes false positives. The molecules are patented for any pharmacological activity because they've never been described associated with any activity before. It has top success in predicting bioactive molecules. It's extremely fast. It's less than 2 weeks for screening more than 150 million molecules. It's an award-winning algorithm in the American Chemical Society Computers and Chemistry Award in 2000, the British K Award in 2017, and we became part of the Fraunhofer Project Center, the first one in Israel in 2019. We have recent success in multi-targeting, both in nanodrugs, loading and stability of nanoparticles, in immunomodulators, toll-like receptors. We have dual TLR7 and TLR9 candidates. In Alzheimer, we have molecules that hit both BACE1 and gamma secretase on one hand, or BACE1 and acetylcholinesterase. In SARS, we have molecules that hit both ACE2 and spike, and some that are candidates for hitting all three, both ACE2, spike, and the 3C-like protease, which is in our cells. And there are 3 startups that are based on ISE, Pepticom, which constructs smart peptides, Rebiotics, which deals with nanomupirocin and nano drugs, Sensotrade, which was a completely different project dealing with the stock market. And I'll thank our supporters in Israel on the first line, the European support on the second line, and Novartis and Bill and Melinda Gates Foundation. And I thank you for your attention. And this is the nice group. Here is Shaima, who is the central figure in the project that I presented here. All the rest of the projects are mentioned here. Anas with cancer, Omri with epilepsy, Noah Stern, Dr. Noah Stern with Alzheimer and with the immune system, Yasser Khatib with iron chelation, Shaima CNS, and now she has a project on artificial sweeteners. Alex looked at fertilization with support from the Gates Foundation, and Daniel is looking into glycogen storage disorders. Thank you.
Thank you, Dr. Goldblum. I think that team is really lucky to have such an amazing leader like yourself. I was impressed. I worked at Teva Pharmaceutical, so I was one of the guys educating American physicians for Razagilin, milnacipran, when you mentioned MAOB, on behalf of Teva Pharmaceutical in North America. So it's so interesting to see the in silico discovery side and the amazing, wealth of information you're providing. I really appreciate and for your leadership and amazing presentation. Thank you so much. It was one of the best I've ever listened to in silico discovery for many years. Thank you.
Thank you.
In attention of time, I would like to, if there are any questions for Dr. Goldblum, I would like to get those questions right now. This is the best timing to receive questions because, you cannot find this amazing gentleman live at this stage. So, any questions about the amazing presentation Dr. Goldblum has provided?
Can you hear me?
About in silico discovery. You're welcome to unmute yourself and pose a question. Thank you.
Hello, can you hear me?
Yes, we can hear you. Please introduce yourself and ask the question. Thank you.
Hi, I'm Akash, and I'm from Italy. And I have a question. It's not a question, it's just a curiosity that if I have missed in your part and you have mentioned, I'm sorry about that. But I would like to know what is, have you targeted to also pass your studies through molecular dynamic simulations to see protein interactions with the compound targets?
I'm not sure I got there. I'll try to repeat the question and let me know if I'm. Are you asking if this method could be applied to molecular dynamics simulations?
Yes, using molecular dynamic simulations was
Okay.
can solve protein.
We do not. We try to avoid molecular dynamic simulations because first of all they're extremely lengthy. They can provide lots of information, lots of important information especially about the protein conformations and the flexibility of proteins and so forth. We dealt with flexibility of proteins in a bit different way. Actually, this is one of our first publications in PNAS in 2002, where we looked at side chains. We looked at the rotamers of each side chain in a protein, and asked the question: which of those conformational states of each and every one of the side chains it has better energy has lower energy than the others. So we created a sort of a Boltzmann distribution of conformational states of a protein by looking at problems of the size of 10 to the 100th power, because there were so many side chains and each of them had many rotamers. And so since the whole idea of ISE is built on the basis of having a set of variables, a large set of variables, it could be hundreds and thousands of variables, and each variable has many possibilities. So it's a lot of variables and a lot of states, or a lot of values of each of the variables. So all the combinations are the number of possibilities, the number of possibilities for each variable, the number of values for each variable to the power of the number of variables. So it's a huge number. We compared our results to experimental X-ray results. So this is not a case where we could compare to molecular dynamics results. But we did compare to molecular dynamics results with a different problem of looking at the dipole moments of small molecules. So what we did is the same as I told you now about proteins, we did with small molecules, where we looked at all the rotatable bonds in a small molecule with, I think, something between 4 and 15 rotatable bonds. And we asked whether we could calculate the dipole moment based on the Boltzmann distribution of the conformations of each molecule. Now this was compared. This was published in a journal of physical chemistry a couple of years ago, where we showed that when we compare our results from just a conformational analysis compared to molecular dynamics with the same force fields as we did for the conformational analysis, then we get better results, better comparison to the experimental dipole moment, which gets its contributions from different conformations on the Boltzmann distribution. So the lowest energy contributes more to the dipole moment, and then there are, in some cases, there were 10 or 15 different conformers that contributed to the total dipole moment. And the more conformations that we used to introduce into the dipole moment from our ISC distributions, we were closer to the dipole moment, and we were overall closer than molecular dynamics.
Thank you so much. This was really a great opportunity for me, for a young researcher, to get such an enlightening thing. I really appreciate it. Thank you so much.
Any other questions for Dr. Goldblum before we move to Martin Emanuel Bittner in United Kingdom? Any other questions? Thank you so much, Dr. Goldblum, for the amazing introduction and amazing presentation in silico discovery. We are grateful. Since there are no more questions,